So please feel free to pop things in the chat. We'll manage them that way. And we can be interrupted too. We don't mind that either. If you want to raise your hand, whatever is easier for you.
Yeah, and we'll record it because it's kind of...
I will get started.
Sorry.
Go ahead, Maria.
Just that we don't normally record focus groups, but we will record this one because it's kind of a tutorial session. But please feel free to speak up.
Definitely. All right, super. All right. Zoom folks and screen good. All right, so I'm going to get started with a little bit of a very short intro. Really, today's session is about making your data and your results accessible and, more importantly, useful. And this is a focus session, as Maria just said. So I really want to, allow you afford you the opportunity to chat and, give your feedback as we go. So I want to first dive in with a bit of context. And actually, one thing I want to do first is introduce some members of our team. We have two members of our team that joined the group recently, Drew Height, who's here, and UIG. Both two members of one is our member at ABI and one's a member at the Broad. We're very happy to have them as part of the team at the Broad. So I just want to welcome them if they're on the call, but welcome them because a lot of the work you're going to see is contributed by them and all the others you see here. So let me get my slides advancing for us. All right, so back to the theme, which is that you want to make your results and data accessible. Really, that's what the Type 2 Diabetes Knowledge Portal was founded on many years ago, back in 2015. The whole concept of that portal at the time was to make data and results, more importantly, from genetic association studies of diabetes and related traits accessible to folks who don't have that ability or aren't computationally savvy and just want to know what are the results of these large-scale human genetic studies. And that's really what the core principle of what the portal was founded on is. And, it's very timely for what we're going to be talking about today because many of you who may be applying for NIH grants noticed a big change. And our field's been moving here, right, for the past, 5 or 6 years now, which is to make all results from the studies we all conduct accessible to the broader scientific research community. And NIH is sort of enforcing this with their new data management plan. Seems a little, daunting at first if you've never done this, but what's really nice about this is the template's really great by the NIH. It's really straightforward. But what's really cool is that there are some NIH repositories that allow sharing of data, and we happen to be one of them. And that's really nice because if you go there, you can use the AMP-CMD portal as a place to deposit your results. And so we're already part of this process. And I think that's a very cool thing for you to think about as you're making these data management plans. And also, if you have any questions about making them, since we've been doing this for many years now, we are happy to work with you as you make your plans for your NIH grant, just as a sidebar. But as a result of what we did with the Type 2 Diabetes Knowledge Portal, all of the platforms we build using using this software infrastructure are part of this repository structure. So you can use this as a place to deposit your results and data. This holds true for the AMP-CMD-KP and the Association to Function Knowledge Portal. However, and in addition to all of our community portals. But what's nice is, in many cases, you have simple results, right? You want to make your results accessible to a broader scientific community. And you don't need a giant portal, which we built for diabetes and cardiometabolic disease, to do so. So what we want to create is sort of a novel way to make your results accessible, fast and easy. And this came about because, a lot of the genetic studies were also going to have new types of genomic and genetic data that needed to be accessible and really had large-scale file formats that, if they're embedded in papers, you don't really get to see them and interact with them. So things like cluster analysis. This was our first one where we had these beautiful heat maps by Miriam Udler and colleagues and a very large table. And we had to figure out how to make that accessible so that a researcher can click through it and see the results and see the underlying statistics and interact with the actual static tables that you'd see in the paper. So this was sort of our first, gap that we tried to fill with a resource. Additionally, and this is really profound, so many of the genetic association studies are now moving to not just doing GWAS, but also layering other types of genomic annotations and bioinformatical approaches that they're integrating into their results and making these very complex tables to produce what we call potential effector genes. And this is one example from kidney disease. We had a list of 761 potential effector genes, and you had all the underlying evidence and support for these predictions in this paper. But you could only get access to this if you had a subscription to Nature Genetics, and if you, for example, were dug through the 28 supplementary tables. And this is what they look like. many people don't want to work with these, right? They're not very accessible, they're not interactive, and they're really hard to read. So we came up with the approach of how do you turn this into a service by which someone could actually upload the results very simply and produce a set of pages, figures, visualizations that are interactive, can be submitted as part of a journal or on its own as a standalone portal, or ideally integrated into these broader portals that we've been building for complex diseases. Additionally, people had also complex visualizations that we had never figured out how to standardize. So we had another challenge here where people wanted to make this interactive. How do you do that? So we came up with some guiding principles. The first is really, at the end of the day, we want these results to be presented in the way you intend as the scientific researcher. We want to produce a set of standardized visualizations and inputs, but the ability for you to customize those to the way that you might like your results presented. So standard things in the field that are used, heat maps, volcano plots, FUOS plots, things that are standard in genomics and genetics, but making those customizable so that you can tailor them to your results. We wanted this to be a collaborative activity such that we're working together with the researcher to, onboard different types of data and determine what types of questions or ways you want to interact with visualizations such you can make them accessible and useful. It had to be easy to use. It had to be such that someone else could try it. If you're not computationally, comfortable with software, you can actually just upload your own flat file and get a portal produced. And finally, we wanted to allow staged access. So you could be protected or open if you wanted to, for example, just submit your portal to a journal for review and then make it open access if it got published. So that was another core component. And then finally, because we've been doing a lot of this work in human genetics and genomics, we have all these results and data sitting in a knowledge base. And what's great is if you have your own GWAS statistics and you want to layer them with genomic annotations that we've incorporated into our resource, we now have made that possible. So you can leverage all of the data and results in the HuGent research or these common metabolic disease portals as part of your visualizations as well. These guiding principles, I want to sort of walk you through that and set the stage for what Maria and DK are going to talk about as to how we implemented this into a software service, what you can do with it, and some of the use cases that we've been able to move forward with our collaborators in the field, and answer any questions you have, and also welcome you if you're interested in building any of these byor portals, as we like to call them, bring your own results with us and making them accessible. So with that, I'll turn it over to Maria and DK to take us through the rest of today's conversation.
Okay, great. Thanks, Noelle. That was a great introduction to set up what we're going to talk about. Can you unshare so we can share?
There you go.
Okay, so move these windows out of the way. Okay, so I just want to kind of give you an overview of what you can do with byor and how you can get started with it. And I will attempt to give a live demo to show you how easy it is. And then DK will talk about some more of the fine points. So to the landing place for this service is byor.science on this page. And you'll find links here to everything you need if you want to try byor. So I want to first call attention to a really great tutorial that our engineers DK and Annie have put together. And it's really comprehensive. It takes you through all the steps you would need to do to upload your results and make a nested table of them, add visualizations, add filters, and so forth. This really, it's a great tutorial. So this is a good resource if you want to get started. Also, you'll need a user account to get started, and there's a form you can fill out, but you can also just email me. That's fine. I can add you as a user. And so there are many different visualizations you can use with byor, and I'll show you some in actual portals. But let's see, why is the demo portal not loading?
Oh, yes. So you need to refresh the page because now I updated the server recently. Yeah.
Okay. Okay, that's a good thing to note. If you're using byor and nothing happens, just refresh the page, because yes, DK updates this every single week. So there are, let's see, there's something in the chat. I just want to see if it's a question.
I'm answering it if you want to
Oh,
go.
okay. Thanks, Noel. Okay,
Mhm.
I'll continue because I think this is not specifically about byor. Okay, let me just quickly show you some types of plots you can do. You can make a Manhattan plot that represents your results. So this is going across the chromosomes, and you can plot scores or just values of any kind that you want with a Manhattan plot. Volcano plots. And all these pages show you the code configuration that you will need to make these plots. So that's really handy. So here's a volcano plot you can make. You can make a heat map. This might be a big one here. Oh,
No, just refresh the page.
okay. You can make a heat map like this. You can make a region plot that shows associations across a genomic region. Oh, and here we'll have to, actually, I won't show it here. I'll demonstrate this with another interface. Okay, so this is just all part of the tutorials here. I want to show you some examples now. Let's go back.
I would suggest to the Dr.
Yes.
Ross or Dr.
Yes, go ahead. Cheryl, was that you? Yeah, I said you have a question. Please go ahead.
I think you're muted.
Did you mean me?
I heard a voice, and I'm not sure from whom it came. I thought I saw that Cheryl's window had a green thing around it, but if someone has a question, please go ahead.
No, I think somebody tried. I'm in a public place, that's why I cannot show my camera. I think somebody tried to answer it. Like I've been doing some search using the metabolic disease portal, but I was not always sure whether if I put in a gene or a SNP, the results I get are exclusive. Like you see phenotypes and other things that are associated with the gene or the SNP. And I'm wondering whether are those the only traits or associations with the SNP or the gene? Like, let's say something related to cancer or something, you wouldn't see that. You wouldn't see results for that, right?
Yeah. Yes,
Yeah.
I was actually answering in the chat. This was your question, right, David? Yes?
Yeah, I think you answered it in the chat.
Yeah, I was continuing, but in short, from the metabolic disease knowledge portal, you will see associations for most common metabolic diseases. That's not to say that if that variant or SNP was associated with psychiatric disease, you may not see its association in that particular portal. However, we just recently launched the A2F KP, which is a2fkp.org, which attempts to address common disease. So therefore expands us to about 500 traits right now that gets into the concept of, is this variant or gene associated with common disease, and allows you to search across a host of common disease. And this is obviously something that we're working with the GWAS catalog and other large scale resources to incorporate as many common diseases as possible. We sort of have a principle for how we define a common disease. But for example, cancer right now is actually not addressed in our knowledge portals at all. Any of the germline cancers. That's something we've had an interest in. But so the answer is, if it's not a trait that we have results for, you won't see the results, essentially. So you can easily search with traits and data we have in the portal right when you drop in, so you can see what associations for which you will get statistics. And you'll be able to get statistics for all of those traits, even if they're not genome-wide, if you dig deeper into the pages. So we're happy to talk to more about this if you want offline, because I think I want to make sure we focus on today, but I'd be happy to share with you and give you a tutorial at some other point if you want.
Yeah, I want to stress that for anyone who has questions about the portal, please just email us help@kp4cd.org. We're happy to answer questions anytime. So we have a raised hand.
Hi, sorry, can I ask a question?
Sure.
I missed the beginning part. So the input is a gene list with the differential gene expression p-value when you mention the volcano plot and the heat map.
That can be, but this is very versatile. So you can really input a lot of different types of biomedical data, as long as it's in a comma separated values file, or if it comes from an API. So it doesn't have to be genetic associations necessarily.
And you have different like a screen to gene. Are you going to go through different parts, or maybe I missed it?
Yes,
Like,
I'm gonna talk about this.
Thank you.
Just about to.
you.
Sure.
What-
So on this page, we list some interfaces that external collaborators have made with using byor, and also these top two are portals that we have made and are connected to the knowledge portals, but also using the same byor system. So let me just show you briefly some portals that external investigators have made. For example, in this study, some collaborators were looking at variants that were reported in ClinVar, and they were looking at different aspects of these variants, such as the coding changes that they made and so forth. And they had just a lot of data, and this was accompanying a publication of theirs, and they wanted to, make this available for reviewers and then eventually for the public. So they made this nested table, which just for each variant includes all of the relevant information about that variant. So this is a pretty simple example of it, and the table can be filtered by many different criteria to find specific variants in the table. Another example of something that a external collaborator did is a really cool site, and this is not only it's not just one byor page, it's basically a whole portal that he made, and byor has this capability. So you can add multiple pages and link them with a header menu like this, and then have different interfaces. And in this case, there's the interface has a gene search. So you can put in a gene name and get information about that gene. And then there are even links to other interfaces where you can search for eQTLs within certain ranges of this gene. So I think you can see that this is a really versatile interface. I also wanted to talk about how we have used it internally in the portals. So as Noel mentioned, we are collecting predictions of effector genes from multiple different disease research communities. So for many different diseases and traits, these are lists of genes that are sort of expert curated, and this is like the gold standard list of genes that are important for specific diseases and traits. So we're collecting these lists and making them available. Yeah, as Noel said, they're often buried in the supplementary data. They may not be open access. And I'll show you a quick example of the paper that Noel showed, the study from Katalin Šestak's lab of kidney function effector genes. So that huge complex table that Noel showed from their supplementary data, it's still complex, but it's distilled down into a kind of easier to work with form. And so these rows show all the different types of evidence that they considered when making these predictions of kidney function effector genes. And again, you can filter the table to find the gene you're interested, or set a threshold for the gene, the priority that you want to look at.
One nice feature is that if you build any of these, and DK will talk a little bit how you're going to build these, you'll see right here, you're going to see the gene name, ALKAL2. You click on that, it takes you to the portal, right? So it takes you to the gene page. So it allows you to link it to the broader resource, which I think is kind of a cool feature. Moreover, as we build these things out with you as collaborators, we can link to more things, right? We can incorporate it into other modules in the portal as needed, or we can link out to other resources where the data may be stored. For example, if you use Geo or ENCODE for anything, we can link out to those things and create sort of like a web, if you will, a network of where the results are buried, like the actual raw data, for example. So that's one thing that you'll see later, I think, for example, when we show annotations. Annotations that are built into some of these are hosted in the CMDGA, which is our sister resource that houses all of the genomic annotations, RNA-seq, bulk data, single cell data, all of the perturbational results and things like that. So you'll see this as we go through, but it's that linking to the other resources and the leveraging of the deeper data that you can use with the byor tool on top of just making these tables interactive.
Okay, let's see. I'm going to go through a few more examples. This is a very new thing. It's not even publicly available on the portals yet, but it will be. I guess it is publicly available on this page. So we're just starting to do this. We're starting to collect lists of genes that are from screens of various types. So these are not effector predictions, but so one type is differential expression. For example, here's a new study where the investigators looked at genes that are differentially expressed between pancreatic islets from people with type 1 diabetes versus people without type 1 diabetes. And another type of list would be gene perturbation. And this is a recently published study from Anna Gloin's group where they did a CRISPR screen to knock out genes and see which knockouts affect insulin content. And so I'll just show you quickly some examples of this. So here's the differential expression, and we've done a volcano plot here. So and there's also a table, so you can see where the genes are. And you can filter this, both the plot and the table. If you're interested in a particular gene, let's see, I'll just put one that I can see here that is here. Oh, did I mistype it? Anyway, okay. If you put a gene name in here, it should, and actually this is, okay, there's something wrong here. And I'm the one who created this.
It may be your range. It may be your range of p-values. Does it?
No, when you put in a gene, it should filter. You should see this point, and you should see MDK in the table. But this is okay. So I think.
Are you on dev?
Am I on dev? No, I'm not on dev. Should I be?
So, Maria,
Mm-hmm.
can you try just MD and see what happens? Yeah, so I believe since there was only single single gene,
Mm-hmm.
so it just plot didn't know how to handle that. But the table must show only MDK if you search for MDK.
No, actually, so it's bringing up other genes that have MD in them.
Yeah, because you search
Right.
for MD. If you search for MDK, then the plot may not know how to handle the single gene.
point.
Yeah.
Got it. Okay. Okay, well, good. Okay, I'll show quickly the view of the genome-wide CRISPR screen results. So here, for each gene, we've got they either caused an increase or a decrease in insulin content, and then there's a value for the mean log-fold change in insulin content. And so this is all here. The graphics show you really at a glance the magnitude of the differences between the knockouts of each gene. And again, this is all filterable and sortable. All right, let me show one more example of how we've used this, and this is the most complex example, the variant sifter tool, which if you've been to these focus groups before, you've probably seen it. But this one, I'll just do this. Okay, it's doing this example search. And I'm not going to go through this in detail because I've done it a lot, but with this interface, so it's showing an association plot across a region. It allows you to select credible sets in the region and epigenomic annotations. And as you go, it filters down to give you a list of variants that answer all of your criteria. So it's a way to really look in depth at a region, at the genetics and genomics of a region, and to prioritize variants and genes in that region. So now I'm going to try to do a demo. I feel like I'm kind of stepping on a tightrope here, but I will do it to illustrate how easy it is to actually do this. And I am the opposite of a computational person. So really, if I can do it, anybody can do it. So once you have an account, you'll come to an interface like this. And if you want to add a research page, just do this. And so let's call it focus group demo. Let's call it focus. Okay. And then we need to upload some data. And just to save time, I've already uploaded some data, but let me show you what the data are. Well, here, this is the file. It's already uploaded via a simple upload interface, but here's the file. So this is actually the raw data from one of those effector gene lists that we have. And these are also kidney effector genes, but they're from Stanzik et al. So in their supplementary data, they had this huge table. You can see I'm scrolling across it. It's really, really big. It's hard to read. And so this is the table that we started with. And so this table is what I just uploaded. It's a comma separated values table. Now that is here. The data type is CSV. There's lots of stuff that can be filled in here, but I'm not going to do it right now. So I'll make this public. I'm just going to save this and we can see what we get right away initially. So just have to, this is not it. I'm just substituting the URL. Whoops, that was focus. So here's that table. So it looks just like it did in the CSV, but it's there. So you can see it's very easy to upload these. And the next step is configuring the table, and it's very versatile. Let me find my code here. So this is more complex. If you're not a computational person, you have to put in some JSON code, and I am copying and pasting some here. And I'll kind of explain it to you a little bit. It's for those of you who use JSON, this will not look like anything difficult. So basically, using this code, you can set the names of the columns that you want to see. So your raw data may have some name that is not user-friendly. You can set a user-friendly name for each column. That's what all these rows are. You can also do conversions. Let me see if this has any. I guess this doesn't have any, but using this, you can add links. You can combine two columns. For example, if you have like a column for chromosome and a column for position, you can make them appear together in a column in your final thing. And then you can do the structure of set the structure of the table. So you have top rows, and then you have some feature rows, like this row with information about the locus, about the variant, about the QTLs, and so forth. So I'm going to save it with this configuration, and we can see how it changed. It's a big table, so it takes a few seconds to load. Okay, so now we have a much more manageable table. It fits in a browser window, and the supporting information is organized in a logical way. We could also add filters to filter this table any way we want. So, oh, I'm relieved that actually worked. And I see we're accumulating a bunch of chats here. Are there any questions relevant to this? I'm not sure.
I've addressed a couple of them, Maria. I think we're in good shape.
Okay, good.
But feel free to, if I'm not missing anybody, feel free to pipe up and tell me.
Yeah, please speak up, anyone, if there anything about that, if there are any questions about it. Okay, I think that was basically what I had to say, and now DK is going to talk to us some more about how byor is used.
All right, let me share my screen.
So you're on here. I might, while you're showing your screen, great stuff. I'm kind of new to this, but I am the principal investigator of StarNet. We kind of joined a cardiometabolic database on multiple tissues. I mean, I guess this is a general question, but the way I see it is that first you go into the portal, and then you would like to select the number of, you like an overview of what kind of databases there are. I don't know if this is even possible, but then you select the database you want to query, and then from there, you have all these nice tools for different visualization or data analysis. Is that, I mean, now we talked a lot about the genetic variants, but also for differential expression, or is I mean, I guess that's kind of the focus analysis you showed, Maria, or did I misunderstand that? Is that possible to actually do those kind of sort of sub-selection of different kinds of, you know, I'm kind of thinking of functional data. I don't know if that.
Yeah. So the interface that I showed, the variant sifter, lets you browse and select functional data. Yes, from different tissues. Yeah, sorry, I didn't go through all of that, but you can definitely select to see certain types of epigenomic annotations in specific tissue types.
So you can, for instance, okay, I want to see whether there is, you know, you can go maybe have an organ focus and then get, you know, the data that are available for the liver, for instance, and then
Yes.
work usually.
Yep, you can do that.
Because yeah, I think just for, you know, I like cross validation for you know, different purposes and generation of you know, novel manuscripts and so on. That would be wonderful. Yeah.
Great. I see Eva has her hand raised.
Okay, so one of the interesting things recently is via Twitter, there is a woman who's an MD and PhD that was explaining her research into a particular biochemical species, and it turns out it's the same biochemical name with different versions in all the tissue types in the body. So what I am hopeful of with the Broad Institute incredible portals is eventually people will be able to get in there and put in something like FGF-21, which I think is the name they gave it when they first discovered this biochemical substrate. And then just as his question asked you about sorting with various computer programs, you would be able to get into the portal and put FGF-21 and then put liver, and that would get you the research. Whether it would give you the actual papers, which some of them might be in peer-reviewed journals, is a different concept. Does that make sense? And I'm hoping DK understood my question. So if not, ask me one back.
Oh, sorry, no, I was thinking about something else. I mean, I thought, your question is related to science, so I thought it's for, Maria.
Well,
sorry about
maybe,
that.
No, but what it is what she found was this same biochemical, which we'll call FGF-21. A different version of it was found like in the liver and in the kidney and in the skin or muscle. And so what we're going to end up having to do is do a sort by the names of, we'll call it, does that make sense now? And so in order to, we'll come up with genes, and it seems like, well, gosh, this gene is changing how the liver and kidneys work. Well, that's partly true and partly not. And that's because the DNA in every cell, some of it is for the kidney and some of it is for the muscle. And the biochemistry is very similar, but different. And that's because, I mean, the basic of it is we all grew from one cell, right? So yeah, so some of the questions that you're going to end up getting is, does this gene do this? And it's not, it's going to end up being, does this gene do this in what tissue type?
Yeah.
So
I'll send an example to Maria is what I'll do, because she'll get it when I send it to her.
Sure, that's fine. Thank you. I think the short answer is that we want to collect as much information as we can that's relevant to all kinds of disease, and it's just a question of how to get it, how to organize it, and how to display it. And we're always
perfect. Thank you. And I think that was a question somebody asked you about different computer programs they're using to sort down biochemical results of genetic research, correct? Someone asked in the chat a question, and I think that's what their question. I'm a big picture person. I try to figure out like what is his question referring to.
Okay, thanks. Please email us more questions. But yeah, let's they go ahead.
Perfect.
Yeah, so I mean, Maria and we'll look at it, from a science standpoint, and I look at it from just an engineering standpoint. So as long as the data is manageable and then accessible, then you can query it by whatever properties you want, and then just sort and filtering. Sorting and filtering is just built in byor, so no, not a problem. Whatever data you have. No, so when we built byor system, we didn't limit the data only in biology, so it should work against any data you have. So okay, so anyway, I will give you a tour to a project we have been working on, which is predicted effector genes, knowledge portal. So it's just still under development, so there's not much other than logo, but we have functional pages and the collection page. So I'll go over those, and then we'll share how we used the data on byor in our portals, so you can get the idea how you can actually utilize byor system. For example, you have a portal, you have a website, and then you want to use byor as a data point. Then you can actually use it as a data point, or also you can actually build your own portals on byor. So I'll give you like the tours for those two examples. So first, we have this portal with 3 pages: home, and then we have this collection page, which has 40 under the pages, sub pages. Then we have a tutorial page, right? And then so let me go to the configuration page for this one, which is this one. If you go to edit, then you can see that we have, so first options include this project to the front page of byor, and then you have data. And in this case, it's a direct input CSV. So you can just copy and copy and paste your CSV data into the data point field, right? And then we have this table format to process the data. For example, it doesn't have too much of, yeah, so we are formatting one of the columns to link field. So we are adding this link to the values in the column. And then, yeah, so here, PMID as well. And then we are adding filters. So that creates this page, right? And then there is a built-in API functionality on byor. So this data in the data point, as soon as you create the configuration page, it is available through API. So you can fetch the data from anywhere actually. So if you have a website and then you want to use byor as a data point, right? And then you can fetch the data from byor and then use on your website. And we have 3 pages we've been using that data, fetch it through this API. One is on our phenotype page, we have EGL section, which filters out the list of predicted effector gene list by phenotype, in this case T2D, and then gives you the list, and then builds link. So you can actually look at full data, full gene list. And then we have gene page, and then it goes over the list and then find the list, predicted effector gene list that contains this specific gene. And then, of course, it has no link to the full list. And then we have gene finder. So here, oh, I clicked the tutorial. So here, you can select the, I'll choose type 2 diabetes, select the phenotype, and then filter it by predicted gene list. And then it gives you a link to full gene list. This case, I think the paper is not actually published yet. So I will try something else. So this one is published, I believe. Then you can actually view the paper from here and then get the full list. So it's built of this API from the byor. And then, oh, this is a variance shifter. And I wanted to give an example of the extreme case, how we can build from of a byor. But since Maria already did, I will just skip it. And then, so this is our future plan. So we found manually inputting, feeding these configurations on the configuration page actually makes some mistakes. So for example, you losing one quotation or in a comma can just break the page. So we are in the middle of building this form that you can actually build configuration pages by GUI system. So you can just select whatever, click, and then by the end of the page, you just save it, then it will build configuration for you. And then will be accessible, right away. So this is our future plan. Any questions? No?
A couple in the chat, but they are a little bit related to slightly different topics. So we'll get to them actually in a second. But yeah, I think any questions for this particular part with DK though? What's nice is, while DK is showing you this, and I think Maria's used it to the point where she's very comfortable with it and she can build pages pretty fast. Pretty cool. We really also welcome collaboration. So if you have like a data set where you're like, you know what, I don't want to configure, I want to call you up and email you and talk to you about it, we'd rather do that. We're looking for new projects. We're looking to host results. Really, that's the goal. It's to host and represent results from genomic and genetic studies and to use that to really strengthen the overall resource, which is like getting to these other questions like representing QTL results. Right now we have places in the portal where we have those results accessible, but what you're hearing about today are researchers who have QTL data of their own that they want to represent on a standalone page. So that sort of fits into our broader portal network, but it's in its own portal right now, or page, if you will. But our broader research framework is aimed at using those types of use cases to develop visualizations that are incorporated into our broader portals. So some of this is a bit of like, helping us develop our roadmap for how we represent, complex trait results and data and genetics and genomic space more broadly, but also really helping you as users to make your results accessible. So, if you feel like, wow, I don't even want to try building my own, just call us up instead of the long way of saying that. Questions?
Could I ask a quick question?
Yeah. Hey, John Paul.
Hey, good day. I saw the information on the eQTL, and maybe this does exist, but do you know if you'll be integrating pQTLs as well?
Eventually, yes.
Okay.
So
okay.
we have visualizations that can handle it. We just need to get the right, expression data for protein data for
Okay,
that.
sorry.
So, yeah.
Sorry for
sure.
for the idiosyncratic question, but
No, not at all. That's a
Yes.
really good question.
We have another question in the chat about EQTLs from Chabot, where she can find gender-specific EQTLs for a gene that she's interested in. And I don't actually know how to answer that.
Not yet.
Yeah, we don't have them in the portals yet. The CMDGA, our sister resource that has genomic information, does have some EQTL data sets that are not yet represented in the portal, but I'm not aware of gender-specific EQTLs. And that could just be my own ignorance.
Oh, thank you, thank you. I have another kind of question. If we use this, for example, the when we use in the paper, do we cite both the website and also original publication? Because you also have like a 2022, 2003, like whichever publication. Do we cite both? How do we cite? If we
cite the portal. Yeah, so we have a publication coming out about the type 2 diabetes portal. That's supposed to come out in less than a month. So that will be a good citation. But on every portal, there's under the information menu, there's a policies page, and that gives information on how to cite the portals.
Use it. Okay. Thank you.
Other questions or thoughts?
So on the note of the predicted effector genes, the reason that we have such an emphasis on those is that there are, Maria's done this really nice curation of the literature to find that there are a host of these out there. And I would say, I think it's fair to say that many GWAS studies now are including this as part of their representation of their summary results, right? So we are actively hoping to work with others who have these lists that they're interested in presenting. We will host them for you, represent them. Ideally what you'll see soon coming into the portal, and I don't know if DK mentioned this, I might have missed that when I was answering a question or whatnot, which is, we mentioned it like a standalone UI for the predicted effector genes, but when you're on any of our portals and you're in a gene page, for example, soon you'll be able to immediately see, if you're on a gene page, you'll see all the genetic associations, the annotations, the variant effect predictions, and all the features that you're used to seeing with our portals. But if you also want, you will see a little, card that notes, hey, take a look at this. This gene appears in 1, 2, multiple curative effector gene lists. So you can immediately go to that list and see the evidence sources. The idea is, we're building up a case for these genes, right? Where many of our users do not want to spend time, weighing through p-values and, and they don't want to go to a locus zoom plot and start with a big region and winnow it down. Some people might want to do that. We offer that capability, but if you just want, I want the list of the genes for, kidney disease or for stroke that the research community for stroke genetics believes are the predicted effector genes, where are they? You can get those, you can extract those, you can visualize those. So that's really, I think, how the field is moving, and also how we plan to follow along with that with the resource that we're building. Any other questions, comments, feedback? All right. And also, if you have any data management plans you need to write for the NIH, I'm serious. I know that they feel daunting, but our resource really does have the capabilities both for the lightweight versions and the deeper weight versions, if you will. So that would be great to talk to you about if you're interested in any help with that.
One last question from Johan is this a bi-weekly meeting now? No, it's every other month we have either a webinar or a focus group. And so it's on the, I think this is the second Thursday of the month. Johan, we do have a mailing list where you can, if you're on the mailing list, you'll get the Google Calendar invitations. I'll check whether you're on it, and I'll add you to it. We also send out, invitations beforehand. So I'll add you to that.
I'd like to add some of my team members as well. They should, I think, being doing this kind of exercise over and over, I think that's how you get used to the site and start using it.
Yeah. Great.
Yeah.
And then in the future, Johan, as your results and data become available in the portal, we're going to invite you back to come talk about your results and how they're integrated in the portal. So you'll become a speaker eventually. So
Yeah, maybe it's, I'm not sure yet because of a visa or a resident status thing, but this meeting in end of March, is it, you know, are there many coming or?
yeah, I can follow up with you offline, but yes, this is a meeting of the folks in the AMP CMD community. For those of you who are not part of the community on the call, yes, you are more than welcome to attend. We can send you information. I think it.
I'm supposed to give a talk and so on. I just wondered, since I'm in Europe, I wonder if I should, you know, it's like if it's going to be like most people there and worthwhile going. You say?
I would say it is, but you know, you are coming from.
You live in Boston.
Yes, right. So it's easy for me. So
okay, thank you both very much.
Yeah, no problem. All right, folks, we'll post this on our site. If you have any follow-up questions, please email us help@kp4cd.org. You can also email any of us on the call. If you just Google us, we're easy to find. And please feel free to follow up with us.
Great.
Thanks,
Great
everyone.
presentation. Thank you.
Bye.
Thank you. Thank you, Maria.
Thank you. Looks great, guys.
Thanks.
Thank you.